biophysicist in the Department of Medicine. And I'm going to sit here so that I can reach my computer, but usually I like to stand and point at things. And today I'll be talking about basics of mitochondrial genetics and some clinical correlations. I urge you to please interrupt me and ask questions and be curious, because I'd rather have a conversation than a lecture. It's more fun for me, and hopefully for you too. So let's just talk about some basics of mitochondrial structure. A lot of this I'm sure you're already familiar with from other classes and from your undergraduate work. But there's just a few important things to know about mitochondria as important now. The first thing is, while we're all used to this very nice discrete picture here of this meeting, this is not really what mitochondria look like in vivo. They're really sort of this fluxing network of sort of these mitochondrial tubules that are constantly fizzing and fusing and communicating with one another. This is just the original image of what we thought they looked like because of early EM photos. So we kind of go based on this to talk about how to contextualize the biochemistry of the mitochondria. It is a double membrane organelle. The outer membrane, the outer mitochondrial membrane, basically its job is to just keep big stuff from getting in and keep big stuff from getting out and make sure that that process is selective. When we think about sort of the small molecule or the electrolyte environment of the intermembrane space here, it's fairly much the same as the cytoplasm. It's just the big stuff getting in and out. The intermitochondrial membrane, which folds into these cristae, that's where a lot of the business end of the mitochondria take place. So that's where the respiratory chain sits. And this folding really helps to allow a lot more of that enzymatic action to fit in and take place. There's a whole lot else that goes on, but the respiratory chain is sort of what we think about as one of the key enzymatic systems on the intermitochondrial membrane. Also, what's really quite important on the intermitochondrial membrane is the machinery that initiates the process of apoptosis with cytochrome C, and a lot of the machinery that's involved in fission and fusion. There's increasingly a lot of human disorders that are associated with defects in fission and fusion that we're learning a lot about. So it's becoming an increasingly focused upon set of machinery for both basic science and translational research. And lots of stuff goes on inside the matrix. So this is where the mitochondrial DNA is hanging out, where transcription and translation of the mitochondrial DNA hangs out. A lot of biochemistry, so the citric acid cycle, fatty acid oxidation, some amino acid biosynthesis and metabolism, among a number of other things. So the reason I put this slide in here is because it's even a joke with my kids, even from when they were little. Oh, the mitochondria are the powerhouse of the cell, which they are, right? That's where the majority of ATP comes from in the cell. But the mitochondria are really responsible for so much more. And so when we think about disorders of mitochondrial function, thinking about mitochondrial function, thinking about mitochondrial diseases, it's a lot further beyond just disorders of the respiratory chain. So I want to emphasize that the citric acid cycle, the Krebs cycle, which is really a nexus of anaplerotic substances that supply carbon chains throughout the cell, occurs there. Of course, we talked about fatty acid oxidation, management of reducing equivalents inside and outside the mitochondria. We talked about apoptosis, and from a medical standpoint, it's really important to know that the mitochondria are heavily involved in ammonia detoxification. You get too much ammonia in the body, it's really bad for the brain. And in the liver, the mitochondria is responsible for part of this process. This slide here is to remind you that nothing exists in a vacuum. And a lot of the biochemical processes that we're speaking about occur within the mitochondria, but really flux in the cytoplasm and back into mitochondria. So we talked about reducing equivalents, of course, phosphate groups, lots and lots of other things that are going in and out. And why this is important to contextualize in terms of both what you're doing in the lab, if any of you are working on like sort of cellular molecular biology, or from a medical standpoint, is that there's lots of things that can make the mitochondria not work that are not inherent to mitochondrial genetics. Does that make sense to you? So if you think about, I think you'll probably hear later in the week about epigenetic disorders, if you haven't heard about them already from Dr. Farner, but that's how genes are turned on and off basically in the nucleus of the cell. But you can imagine that in those disorders, so much stuff is going on and dysregulated that it actually secondarily affects mitochondrial function. So in terms of my medical practice, prior to the onset of the availability of next generation sequencing and whole exome sequencing, which is really only about the past 13, 14 years, I had lots and lots and lots of patients who were diagnosed with mitochondrial disorders because their biochemistry looked really weird, they were making lactate, they had energy defects. But when we were able to go through and actually perform whole exome sequencing on all those patients, a percentage of them did have primary mitochondrial defects, but a larger percent of them actually had other genetic disorders that cause broad defects in the cell. Does this make sense? So it's when we think about mitochondrial function, it's really important to contextualize it. Okay, if there's one thing to take away from today, which I'm again assuming many of you know, but the mitochondria, the genes that encode the proteins that involve the function and the structure of mitochondria come from two places in the cell, two places. And lots of times when I talk to families and I say, oh, we're thinking of a mitochondrial disorder for your child, the mom always says, oh, I knew it was my fault. So first of all, genetics are nobody's fault. But second of all, these people are thinking of this circle of mitochondrial DNA, which is inherited via matrilineal pattern. Right? So there's 37 genes there that are only passed on through the mom because they're only passed on through the egg. But there are hundreds and probably more than 1,000 other genes in the nucleus of the cell that contribute to primary mitochondrial structure and function. And disorders in most of those genes follow an autosomal recessive pattern. So most of the mitochondrial disorders that we see in terms of numbers of disorders, N of disorders, are actually nuclear and autosomal recessive. Conversely, the most common mitochondrial disorders, which I'll talk to you about with the highest incidence, are actually in those 37 genes there. Okay, so two places in the cell, nucleus of the cell, mitochondria. Any questions there? Yes?
What is like the incidence of mutation for base pair in mitochondrial genes? Why are they more common?
Why are they more common? So there's a number of reasons for that. The first is there, and I think I'm going to get into it later. I can't remember what's in this lecture versus the thousands of other minor lectures I do. But so there is a higher mutability in the mtDNA. The first reason for that is because the mitochondrial DNA is basically sitting right up against a lot of oxidative stress. So they undergo essentially a lot of stress as a complex molecular structure. The other reason is while there is some proofreading capabilities in the replication machinery of the mitochondrial DNA, it's a lot less than the nuclear. So we used to think there was no correction machinery. There is some, and I can't tell you exactly what those rates are. So there does seem to be a higher frequency of mutational loss correction. The other thing about it is, and I'm going to get into the concept of heteroplasmy, but the pathogenic mutations can hang out in this mitochondria undetected in the family for a really, really, really long time and only appear if heteroplasmy, which I'm going to get to, becomes high enough. So it's estimated that actually 1 in every 200 people, if you do deep enough sequencing, you're going to find a known pathogenic mitochondrial DNA mutation somewhere in their genome, which is a real bear when you're trying to deal clinically with these things because GeneDx will report out to us heteroplasmy is as low as 3%, and I don't know what to do with that because that's going to come up in 1 in every 200 people, frankly. So I hope that kind of answered your question a little bit, but we'll get into a little bit more of why that is.
I have a question.
Yeah.
So like, I guess just in the nuclear genome, we know like variation is super important for like species. Do you think that variation in mtDNA is necessary or important at all?
In some regards, and I'll talk a little bit about haplotypes, which are, for those of you who might be working with Don Arking, it's a little different than the kind of haplotypes that he talks about. Just so we're going to shift gears a little bit, if anyone's in his lab, shift gears a little bit from that. But so those are sort of lineages that arise from certain ancient populations and essentially govern how coupled the mitochondria is. It's thought to be environmental responses. But beyond that, variation probably isn't as important. Let's go to the next. Can I come back to that? Because there's a slide where I'm really going to get in touch with that. So we talked about causes of mitochondrial disease. And I just want to, again, focus on the fact that there's primary mitochondrial disease, so genetic defects in genes that encode mitochondrial structure and function. Vastly more common, and those can be nuclear or mitochondrial DNA. Vastly more common are secondary mitochondrial disorders. So all the other genetic disorders that can affect the mitochondria, plus lots and lots and lots of other stuff that can affect your mitochondria. So hypoxia, oxygen deprivation, toxins, antibiotics, other medications, illness. The number of consults I get to rule out mitochondrial disorder that are really, really difficult to sort out are situations where there's been oxygen deprivation, and you go and you do all your biochemical testing, and the mitochondria aren't working because the mitochondria are deprived of oxygen. But it is a mitochondrial dysfunction, but not a disorder. So when you go back later, those variations are no longer that they've recovered. Does that make sense? Okay, so the mitochondrial genome is codes for 37 genes. It's about 16.6 kb, and it codes for 13 peptides and 22 tRNAs and 2 rRNAs, 2 ribosomal RNAs. Mitochondrial DNA, circles of mitochondrial DNA are found in every cell of the body except for mature red blood cells. So your red blood cells kind of kick out all their organelles as they grow up because their job is only to carry on oxygen essentially. So they don't truck with mitochondria when they get older. There are some really key principles when thinking about mitochondria, so mtDNA. So I'm pulling out mtDNA from nuclear DNA, right? So these concepts only hold true for mitochondrial DNA. There can be several thousand copies of that circle of that mitochondrial chromosome, that circular mitochondrial chromosome per cell, and hundreds per mitochondria. In general, high energy requiring tissues have more mitochondria, so your muscles, chock full. Your heart is chock full. Mitochondria is probably about 30% of the cell weight for cardiomyocytes. Cells that require less energy, fibroblasts, tend to have fewer mitochondria and fewer mitochondrial DNA, which always makes me suspicious from a basic science standpoint of mitochondrial functional studies that are done in fibroblasts. They do not really reflect what's going on in the rest of the body, and there's been a lot of lessons learned there. So it's an important concept to think about. Usually, mitochondrial variation is inherited from the mother, although occasionally there are some de novo variations that we'll talk about. I'm going to talk about a concept of random segregation during mitosis. Most of the mitochondrial variation, even a lot of the pathogenic mitochondrial DNA variation, does not have a replicative disadvantage or advantage. So when pathogenic variants are segregated during the process of mitosis, it's random, for most of them, not all of them, for most of them. Beyond, above a certain level of mutation load, that's in general when you're going to see mitochondrial dysfunction or disease. And that is this concept of heteroplasmy that I'm going to get to soon. I try to give a little spice for everybody about the mitochondria, depending what you're researching, to make you, excited about mitochondria no matter what you're doing. So for those of you who are interested in epigenetic medicine or epigenetics, for a little while it was thought that mitochondrial DNA was an exception to epigenetic modification. That's actually not true. We're learning increasingly important changes in mitochondrial DNA methylation actually affects mitochondrial structure and function. And there is the DNMT1 transporter transports S-adenosylmethionine into the mitochondria and SAH out of the mitochondria, and there's a new disease that's been recently reported that's actually a defect of that transporter, and it causes really severe mitochondrial dysfunction in children, and a milder form that causes myopathy in adults. So clearly this is important. We're still learning a lot more about it. We do know that there's secondary structure to the mitochondria. TFAM serves to pass mitochondrial DNA into nucleoids. Again, wide open area of study, just something to think about.
Do you know if CRIPS contain a higher concentration of mitochondrial DNA in nucleoids?
I'm sorry?
So it's been reported recently that some subtypes of mitochondria will keep copy numbers in the CRIPS of the CRISPR.
I actually don't know about that. That's interesting.
That's a means of controlling translation.
I haven't heard about that, but if you could forward me a paper, that would be great. I would love to learn about it. I wouldn't be surprised. The mitochondria are amazing. So let's talk about the bottleneck hypothesis. One of the key concepts about mitochondrial inheritance in families is this idea that when a mother is carrying a certain heteroplasmy load of a mutation, it is virtually impossible to predict how her offspring will be affected. And that is due to heteroplasmy and due to this concept of a bottleneck that occurs early in development of the ovum. So when the ovum is maturing, it undergoes essentially a coning down of thousands of mitochondria down to a handful. So we're going from thousands probably down to tens. And it's a random process. And then when the ovum continues to mature before fertilization, it cones back up. And whatever was randomly left in terms of mitochondrial DNA is then what's expanded. Right? So this process is random. And so you can see, if we're looking here, sort of this sort of very false and general representation of mutated mitochondrial DNA versus not mutated, you can see how a mother who has, more wild-type mitochondrial DNA as compared to mutated could have an offspring that has completely severely affected based on heteroplasmy alone, moderately affected, or essentially unaffected. So very, very, very tricky in terms of counseling a family and helping them make decisions for their future. This also can help you understand, with your question earlier, how these things can just, or relates to your question, these things can just hang out in a family forever and ever and ever, until all of a sudden they appear in a generation. If this situation just keeps happening again and again and again, you often don't detect something in a family until this situation happens. What makes this even more complicated, the concept of heteroplasmy, is that I talked about random segregation during mitosis of mutated versus wild-type mitochondrial DNA. That actually also happens as the embryo and fetus are developing. And depending on what mutation load gets into different organ systems, that can actually predict or relate to what organ system is going to be involved. So if we take, for example, siblings who inherited a certain load of mitochondrial DNA, you can look at the first sibling and they got a very, very high mutant load in their muscle, their kidney, their heart, and their brain. And that's going to be someone with multi-system severe mitochondrial disease. But their sibling only got that mutation in their muscle, that high mutation load in their muscle. So they're only going to have myopathy, muscle disease. Or a sibling who only has muscle brain. You can see how complicated this all is. And this is actually a family that I saw. You all know pedigrees by now, of course. This is a family that I saw who came to attention because of this daughter here. Yes, when she was about 11 or 12 years old, she had a stroke. And based on what the brain imaging looked like and her sequencing, it was very clear that she had a mitochondrial disease called MRF, which I think I'm going to touch on later. But when we actually started looking through her whole family, we realized that they all had some aspect of mitochondrial disease, likely based on their heteroplasmy in tissues and what that load was. So she was detected, but when we actually started looking through, this was looking at a stroke, sorry. This patient had diabetes and muscle weakness, migraine, this one had hearing loss, mom had hearing loss. We actually even got all the way out to a cousin who had migraines, muscle weakness, and we actually found potentially the heart arrhythmia in him. So just by detecting her, he didn't know he had this, but when we got out to him, we found his heart arrhythmia. And this is oftentimes what happens, that someone finally comes to attention, and they, the family history you often get is, oh, my family has really bad luck. It's actually not bad luck, it's just a mitochondrial disorder with variable expressivity throughout the family. Okay. Respiratory chain. I'm not going to focus too much on this. Only to know that this is the 5 complexes of the mitochondrial respiratory chain. As those of you who work in my lab know, they don't really sit on the intermitochondrial membrane like this. They're organized in these higher order supercomplexes, and that helps increase the efficiency of ATP production. But it's nice to think of this in this nice little strip to kind of help you straighten out the biochemistry. What's important to know when you're thinking about diagnosing a mitochondrial disease or the molecular genetics of mitochondrial disorders is what are the genetics that contribute to each complex, the subunits that are in there. So complex 1 is the biggest complex. It consists of 47 subunits. 7 of them are encoded by the mitochondrial DNA. 40 of them are by the nuclear DNA. Complex 2 is really interesting because it is only encoded by nuclear DNA. Right? The other ones have components, at least, of mitochondrial DNA. And why this is historically important from a diagnostic standpoint is when the mitochondria aren't working real well, what they do is try to really rev up transcription of the subunits. But if the mitochondrial DNA isn't working well and you look at the respiratory chain function in, say, a muscle tissue, if you see really, really, really high activity of complex 2 and low activity of the other complexes, you kind of start to think that the problem is in the mitochondrial DNA. Does that make sense? So diagnostically, like historically, this was a really important thing, that if you saw muscle with really, really intense complex 2 activity and everything else was down, you start thinking the variation is somewhere in the nuclear DNA. We don't really use this so much anymore with the ability of next generation sequencing. You just sequence through all this stuff and find where it is. But in the days when we couldn't do that, it was really an important diagnostic signal for us. So what I want to sort of emphasize too, and you know, this question may come up later during your oral exams at some point if you have me on your committee, and I say, can you draw me a pedigree of a family that has a mitochondrial disorder? And the question that should come back is, do you mean an mtDNA disorder or a nuclear disorder? Because they really, really want you to, again, there's a few things to take home. It's that mitochondrial diseases, primary mitochondrial diseases, can look like anything. It can be dominant, this could be a dominant case, or frankly, this could be matrilineal, and this patient just really hasn't presented yet, but we'll say dominant. Recessive, which is the most common inheritance pattern. What we have here could either be dominant or there are rarely, one of them we study is an X-linked recessive mitochondrial disorder. Barth syndrome is probably the prime example of that. So you know, you could say, I could draw you any pedigree, and that could be an example of a mitochondrial disorder. A little bit about evolution. Again, I'm trying to give you all the spice of life, depending on what you're interested in. Mitochondrial DNA, all data to this point points to the fact that they do not undergo recombination. That may change, I think, as research moves forward, but right now it doesn't seem like there's that mitochondrial DNA undergo recombination. The mtDNA moves as a single unit, as a haplotype when it's transmitted. This helps us understand evolutionary biology of females, similarly to the way that the Y chromosome is studied in males. We're touching about, I think, Cody, you're going to ask a little bit about mitochondrial variation. The main source of relevance for mitochondrial variation is in following out mitochondrial haplotypes. And these are essentially common variations in certain populations that sort of help us track lineage. And there's some really interesting theories that have come from Doug Wallace's group. So I think he's at CHOP right now, but he did a lot of evolutionary biology looking at mitochondrial haplotype tracing. And if you think about some of the most ancient haplotypes that arose out of Africa, these are the most tightly coupled. But as human migration occurred and we move out to the Scandinavian appearing haplotypes, so haplotype H, haplotype U, these are not tightly coupled, and the idea is that they're actually much more exothermic. So this is a more endothermic haplotype, and this is more exothermic, because your body needs to produce more heat when you live in the cold. It's a really interesting theory. It's obviously impossible to prove, but what's fascinating is certain mitochondrial diseases that we're aware of, so ATP synthase disorders, are actually much more severe when they sit on an uncoupled phenotype, because they're much more exothermic and they lose energy. So there are certain disorders where, depending on the haplotype you sit on, they actually look very, very different. So this speaks a lot to the relationship between the nuclear variation and the mitochondrial variation. All right, so I'm going to move on to clinical aspects of mitochondrial disease, but just to pause here, see if there's any questions, concerns, interests, anything you came across. Yeah?
Why there's no recombination of mitochondrial DNA?
I'm sorry?
Why there is no recombination?
You know, I'm not, so first of all, we're not aware of the recombination machinery that exists within the mitochondrial DNA to correct any errors that occur during recombination, so that's possible. I have to tell you, I personally believe there actually is recombination based on a couple of studies we've done. So do not learn this for anything that you're going to learn other than my experience. There's a couple of patients where we've tried to track whether their mitochondrial, they carry two pathogenic mutations, and we're trying to understand if they're on the same mitochondria or not. And we worked with Mike Schatz's group to do long-read sequencing, and it does appear that in some of these patients, the vast majority of the variants sit on separate mitochondria, but some of them, they look like they're sitting on the same. So I think it's as of yet undiscovered. Does that help a little bit? Maybe, maybe not. Yeah. So I think it's there, but I think there's a lot of them.
The last mitochondria
Oh, sorry. Let me just turn off my render that was saying. Yes, I'm sorry.
No, it's okay. For the last mitochondria, like fibroblasts, like kind of older tissues, do you have like changes in like kind of myofibroblastic changes? Like I guess like is there any kind of like implication in pathology of like this going around like mitochondria or like this kind of changes like that?
You mean as you're looking at sort of different tissue types, cell types, as they develop, as they evolve, and as they replicate? Can you see change? For sure.
Okay.
For sure. So the best fibroblasts in which to study mitochondrial function are fresh ones, because as you get to higher and higher passages, they actually become more glycolytic, less reliant on mitochondria. And there's ways you can force them back in the other direction, but it for sure affects the mitochondrial activity.
How does the cell type influence mitochondrial morphology and, I guess, function?
I'm sorry, I think I need here in progress.
How does the cell type influence mitochondrial morphology? So, like in axons, the mitochondria are longer.
Yeah, that is a really deep question, and depending on the cell type, there's really very different, not only mitochondrial morphology, but also mitochondrial movement. So as you're mentioning in axons, there's a lot more machinery that actually controls the movement of mitochondria up and down those long axons. There's a number of mitochondrial diseases that affect that. So, I mean, I imagine there's sort of an overarching nuclear complex process that controls what's turned on and off, but certainly it's very specific to cell type and the needs of that cell type.
I'm trying to conceptualize if we start with like one single fertilized cell with the same mitochondria, how does it turn into like all the different?
Well, I mean, you can think of that basically for any cell in the body, right? Because every cell in the body has the same nuclear DNA, and it's sort of the patterns in which genes are turned on and off that control that function. And so I imagine it's just as complex as anything else in that. But that's why developmental biology is an incredibly complex process that we're still learning a lot about. But you know, when you think of, we're getting a little bit off topic, but when you think about sort of genes that affect patterning, so defects in genes like sonic hedgehog, right? It affects a very, very specific process and time point in the developmental biology of that child. So for example, a sonic hedgehog defect causes a lot of midline defects, but once those midline defects are produced, whatever's there functions fine. So it's like something that's very temporal. And that's getting on topic. Hopefully that makes sense.
Yeah.
Okay, mitochondrial disease. I'm just going to try to give you a taste of how these diseases can present in people, and how this sort of traces back to the role of the mitochondria in energy production, but lots and lots of other stuff in the cell. But if you think about it, the whole body needs energy. And so mitochondrial diseases can present with any single or combination of multi-organ system dysfunction. And this is why my clinic is very, very busy, and why I never know what's going to come through the door, and why everything is different and interesting and exciting. When we think about high energy producing tissues in the body, I think I'm just going to go from what's referred to me the most to what's referred to me the least, so that'll help. So I get a lot of referrals for mitochondrial eye disease. The mitochondrial retina and optic nerve are very, very, very heavily reliant on oxidative phosphorylation. And so there are certain signals in human eye disease that will make an ophthalmologist think of mitochondrial eye disease. That includes retinopathy, which is like sort of speckling or abnormal pigmentation in the retina, early cataracts, optic nerve disease. When we think about things that go on in the brain, seizures, strokes, but not the kind of strokes that you might think about, which is bleeding of vessels or bursting of vessels. This is actually the kind of stroke that causes tissue death from bioenergetic stress. I'm going to show you a picture of what that looks like. Hearing loss, ataxia, which is an imbalance of walking or using your limbs, and this can come from two places, either your peripheral nerves or from the cerebellum, the part of your brain that makes your body have balance. Heart involvement is really, really common, and as I talk about, it's important to recognize mitochondrial disease because we cannot cure these things, but recognizing them can be life-saving. So I mentioned the patient who, when we finally got to him, tracing out to his family, we found out he had a potentially lethal arrhythmia. We need to recognize, prevent, and treat heart disease. It's really common. Bone marrow failure is a little less common, but in some infant forms. Gastrointestinal issues are hugely common. Muscle weakness and fatigue is probably the biggest complaint. Every patient I see that has a generalized mitochondrial disorder has some aspect of fatigue. Kidney disease, infertility, diabetes. You can see how almost anyone and everyone can get referred to rule out a mitochondrial disorder. In general, a lot of times when I'll do clinical lectures, people say, well, what should be a red flag? In general, it's involvement of two organ systems that seem completely unrelated in a patient at an age when you wouldn't expect it. So, a lot of these things are sort of disorders maybe associated with aging. So if you see someone who's, in their 8th or 9th decade and they have hearing loss and maybe type 2 diabetes, you might not hone in on mitochondrial disease. But if you had a 10-year-old that came to you with diabetes and hearing loss, that should be a red flag. Does that make sense? Now, there are certain combinations that bar none are mitochondrial to proven otherwise. That's liver-brain dysfunction at the same time. And some of these eye findings are very specific.
Is it type 1 or type 2 diabetes? Type 1 or type 2?
Either. Yeah, we don't get away with being able to classify it that way. In general, the type 1 is the younger onset, and type 2 we see in the older adults. And it's a peculiar type 2 because patients with mitochondrial disease generally don't have excess body fat, so it's this sort of weird insulin resistance. It's not necessarily associated with adiposity, but I'm going to talk about mitochondrial disorders in the context of the molecular pathology that causes them, to try to kind of, it's not ideal, but it's the best way I can think to group things for you. So the first 3 groups of disorders we're going to talk about are ways that the mitochondrial DNA can be abnormal in the mtDNA, and then we're going to touch a little bit on nuclear mitochondrial disease. So the names of these disorders are somewhat awkward. They're really acronyms for some of the major features that are seen. The family that I showed you, that child who had a stroke, was affected with MRF, a myoclonic epilepsy with ragged red fibers. Ragged red fibers describes what's here on the muscle tissue, and essentially it's a Gamora trichrome stain, and this red patches here are clumps and clumps and clumps of mitochondria. The tissue is working really, really, really hard to make energy in the face of non-functional mitochondria, so they're just duplicating non-functional mitochondria, and it stains red. I love looking at mitochondrial disease in muscle fibers because this is a beautiful picture of heteroplasmy. Because heteroplasmy, when we measure it in terms of the tissue, right? We take a chunk of tissue and we look at mitochondrial DNA sequencing. But actually, the heteroplasmy is on a cell-by-cell level. And if you look at muscle fibers, which are individual muscle cells, you can see here, this fiber here probably has a lower heteroplasmy for pathogenic variants. So this one has a higher heteroplasmy. And with the ability to do single cell sequencing, this had been a theory for a long time. It's actually been proven. So you can do this cell by cell. So it's beautiful, fascinating. So the 4 canonical features in patients with MRF are myoclonus, or these sort of jerking discharges of muscles, sort of this like jerking energy discharge. Generalized epilepsy, so seizures. Ataxia, I discussed that imbalance of walking. And in patients with this disease, the ataxia comes from both places. Their peripheral nerves, so they can't feel where the ground is when they're walking, and from the cerebellum in the brain, which controls sort of where your body is in space. And ragged red fibers. Almost every patient has exercise intolerant lactic acidosis. This basically means that your body is making energy through glycolysis, not the respiratory chain. It just tells you the respiratory chain is not so happy for whatever reason. WPW is that heart arrhythmia I mentioned to you. Patients who can't make a lot of energy can't always grow so well, so they tend to be a little bit short stature, and I talked about the optic atrophy. But patients with this disease can present with any, all, or one of these features. So actually, the patient only has fatty deposits all around his neck and his chest. That's what lipomatosis is. In my patient, actually, though, it's so severe that it actually caused restriction of his airway, and he had to have a tracheostomy to allow him to breathe. So even manifestations in one single tissue can be life-threatening. So, but you can see why it can be, missed or misdiagnosed or difficult to manage. So what causes MRF? In most cases, it is an A to G substitution at mitochondrial DNA 8344. This is the mitochondrial tRNA lysine. There are some rarer forms of MRF because it's kind of a clinical diagnosis, right? So there's still, after all these years, MRF's been described for probably 30 years at this point. Why does a defect in tRNA, this defect, because there's other defects in tRNA lysine, why does this defect cause these features? Nobody knows. Nobody knows why it does this. Nobody knows why it's so specific. There are plenty of studies to show that there is some disruption of synthesis of proteins responsible for oxidative phosphorylation. But the specific organ manifestations of this disease are still a mystery. Okay, so moving on to MELAS. I think I mentioned to you earlier stroke, right? So when we think about classical stroke, well, I don't know if you think about that yet, the main causes you think about would be a person who has a blood vessel in their brain that either has an obstruction, has a clot in it, or has a hemorrhage, bursts. So when that happens, that part of the brain that's fed by that vessel is where you see the change. That is not what's going on in mitochondrial strokes. So if you think about anatomy of the brain or vessel anatomy, I don't know if you do that anymore. Do you do that anymore? You used to have to do a systems class. Not so much. You don't do neuroanatomy anymore?
We do in the systems, genes, and mechanisms, but in the second year.
Okay, so you haven't gotten there yet. Okay, so there are parts of vessels that feed specific parts of the brain, and they really, we know what they are. They're very well mapped out. When we're looking here at an MRI, I'm not a neurologist. All I know is this is a T2, and white is bad and black is fine. That's the extent of what I can read with MRIs, but that is all you need to know for MELAS. And when you look here, this is a patient who presented with vision problems from the back of the brain and hearing and listening problems, communicating problems in the temporal region of the brain. There is no single blood vessel that is going to feed this and can be obstructed at the same time and cause this problem. And looking at this should tell any neuroradiologist or anybody who knows a little bit about neuroanatomy that this is not a vascular disruption. This is not a vascular disease, right? This is something different, and this is MELAS. So what patients with MELAS eventually causes the progression of their disease is they have basically a cumulative effect of these stroke-like episodes. So sort of unfortunately, once they start, one after another, different brain regions just start having these episodes, and eventually causes really severe impaired ability, and that ultimately leads to death. Why it's also incredibly important to recognize this is that these are not treated the way standard stroke would be treated, right? So when someone comes in with a clot in their brain, you give them clot busters, you thin their blood, you want to make the open up that blood vessel. That is absolutely the wrong thing to do in this situation, because that potentially, you're thinning someone's blood, could cause a whole lot of other complications that are not going to help with this. What are other things that occur with MELAS? Seizures. They can occur with or without the stroke-like episodes. Recurrent headaches, which can be super tricky, because when a patient calls and says they have a horrible headache, should I come to the ER? I don't know if it's a stroke or they just have a migraine. It's really tricky. Exercise intolerance, almost everybody has it. I talked about the lactic acidosis. Hearing loss is quite common. By far the most common cause of MELAS is a 3243 A to G variation in the tRNA leucine. Again, why that is, why it's that specific variant, why it causes these symptoms, I don't know, but this is the class of sort of the prototypical mitochondrial DNA pathogenic variant. So Leber's hereditary optic neuropathy is perhaps the most common mitochondrial DNA disorder, and I think perhaps one of the most interesting, because it's another situation where the pathophysiology is just, it's fascinating. The penetrance is also really fascinating with this disease. So this is a condition where, in general, and I'll talk about there are three canonical variants in the mitochondrial DNA. You have to have quite high heteroplasmy for them. It's usually homoplasmy or above 70% heteroplasmy, which causes bilateral, painless, sudden visual failure. And it can be one eye or both eyes, or one eye goes first, but then the second eye goes later, but it's eventually both eyes. It develops during the second decade of life, so it's usually older teenagers. And for some reason, males are 4 to 5 times more likely than females to have expressivity of their disease. We don't know why this is. The only thing we know that is more likely to make this vision loss occur is smoking. So what we do tell patients, because we do identify a lot of pre-symptomatic patients, because these are things, again, that go through families, is do not smoke, because that's the one thing we know that makes this more likely. So the visual failure that occurs, oftentimes when you talk to a patient, the first thing they'll tell you is they have blurriness. Like it's just all of a sudden they're just driving one day, it's sudden, and the things are blurry, and they go to the eye doctor and they're not sure what's going on. But eventually what happens is they lose central vision. So they get what's called a central scotoma. So they retain peripheral vision, but they can't see what's going on in the center. So there are 3 primary mutations that cause the vast majority of Leber's hereditary optic neuropathy. And again, as mentioned, in general, you have to have a higher mutant mitochondrial DNA load. Now, this is one situation. There's so many patients with this, and these disorders have been so well studied that we do know of secondary variants that can affect, in some ways, the penetrance and severity of disease, but in and of themselves don't cause disease. And we do know that these variants affect respiratory chain activity, but why this affects only the eye, why some of them have different penetrance levels, and what makes this even more complicated is depending on the variation, there's actually a good chance of visual recovery, so which is great. So patients with 11, 7, 7, 8 have about a 30% chance of getting visual recovery, whereas the 1, 1, 4, 8, 4 is probably about 5 to 8%. Which, as you can imagine, trying to do a clinical trial in a disease where a third of the patients are going to have spontaneous visual recovery is a nightmare, because you don't know if what you're testing is because the disease is getting better or because it's the drug. But you are able to offer, it's it's devastating for anyone, but when you're talking to a teenager who comes in, 18-19, who suddenly can't see, you can say there's a chance. There's a chance we're going to get some visual recovery, and let's just see where things go. So now to move on, we talked about point mutations. We're going to talk about mtDNA, mitochondrial DNA depletion disorders. And this is a disease mechanism like none other. So what it is that a nuclear gene responsible for mitochondrial DNA replication infidelity is defective. So you have a nuclear gene causing a mitochondrial DNA defect. The canonical one, or the canonical one, is POLG, polymerase GAN, which was responsible for the replication of mitochondrial DNA. And the disease actually can be caused by 2 mechanisms: recessive, so both copies of POLG in the nucleus are defective, or dominant. The recessive form is absolutely a devastating disorder, which often presents as a lethal neurologic and liver disorder in kids around 2 or 3 years of age. So Alpers-Huttenlocher, if I talk about that in the next slide. I think I get to that. All the way to milder forms. And adults who inherit one mutated copy, can basically have isolated eye problems or external ophthalmoplegia. And I'll talk about that a little bit. The diagnosis for this, of course, you can do via next generation sequencing of the nuclear DNA, but what's really fascinating is if you actually sample the affected tissue, you can detect depletion or loss of that mitochondrial DNA. So you would get a lower percent than what you would expect. Sometimes if you have variations of unknown significance, if you sample in POLG, you sample a tissue, you can detect that loss of mitochondrial DNA. So again, a variety of presentations. Alpers-Huttenlocher is the recessive form. The earliest described, oftentimes the most severe was the first one to be described. It's just, again, a devastating pediatric onset disorder. All the way to sort of, relatively speaking, more mild recessive forms, but I can tell you all the way through the adult onset form of these things, they're still devastating disorders, they just present later. But then there's the dominant form, which is progressive external ophthalmoplegia. So these are adults who lose the ability to move their eyes laterally or up and down. They developed a fixed eye position and drooping of the eyelids, and pole G dominant is a cause of that. So, talk about the next mechanism. So we talked about mtDNA depletion. Now we're going to talk about deletion. So I had mentioned that most mitochondrial variation is inherited from the mother. These disorders are so devastating that they are always de novo. So one thing you can actually tell a family if this happens is it's not going to happen again, because this is one of the rare forms of mitochondrial mutation where there actually is a replicative disadvantage. Okay, so in the deletion, typically the mitochondria itself is actually missing thousands of base pairs. Okay, so we just have a smaller circle of mitochondrial DNA, and it causes a devastating bioenergetic defect. The earliest onset form of this is called Pearson syndrome. So this is basically when that deletion occurs so early in the formation of the embryo that every tissue has it, and the bone marrow fails because it cannot keep replicating. It doesn't have basically the bioenergetics to keep replicating the hematopoietic system. And sort of the classical finding here is what's called sideroblastic anemia. So you basically just get this bone marrow starts pushing out these immature red blood cells that still have nuclei and have iron deposits in them. So these patients, it's usually fatal in infancy. Occasionally a child will get a bone marrow transplant and live longer. We don't have time to get into clinical trials for Pearson now. There is an attempt to do bone marrow transplant and mitochondrial DNA transfer that looks like it's helping these kids with but in general, again, quite devastating disorder. Or Leigh disease. So the reason I wanted to put Leigh disease, or Leigh syndrome up here, is because this is actually a description of when parts of the brain called the basal ganglia or the brain stem undergo sporadic damage, and just trust me where these arrows are here, it's abnormal. Brain stem, basal ganglia, these are abnormal. These are bioenergetic failure of sort of the control regions in the deep brain that control both breathing, movement, and consciousness. And this can be caused by, at this point, there's probably about 30 different genes that can cause Leigh syndrome. So some mitochondrial DNA variation, some nuclear variation, usually in the respiratory gene. What time is this class over?
12.
Oh, okay, we got a little bit of extra time. I won't keep you that long. So diagnosis, increasingly really next generation sequencing is becoming our mainstay of how we make a molecular diagnosis, how we make a specific diagnosis in patients. Because we get it back really quickly, it's really specific, it helps us understand inheritance patterns, what's next for these patients, but there's a number of things, again, considering how variable these things express and how we want to treat each patient as an individual, how we think about each individual patient's diagnosis moving forward. So when a patient comes to me and we're suspicious of a mitochondrial disorder, there's typically a number of things we're going to do to help resolve things for them. The first is, of course, take a really good family history to try to understand, can we ascertain an inheritance pattern, what else is going on with that family. You can do histopathology, so we talked about those muscle studies. I more and more am using them to confirm variations, or if I have a patient where I'm really convinced they have a mitochondrial disorder and we haven't found something on sequencing, we'll go do sort of invasive procedure and look at their muscle and see is there anything there. Biochemical studies, I talked about lactate. When lactate is up, it means glycolysis is up, oxidative phosphorylation is down. There's other things we like to look at that also tell us that there's oxidative stress, that the mitochondria aren't doing their biochemistry the way they should, to be sort of prosaic about it. So organic acids, plasminic acids, and molecular studies. So in general now, unless someone walks in the door and I know what they have, and I know it's MELAS or whatever I think it is, I will send mitochondrial DNA sequencing, but more and more I'll actually send the whole thing. I'll send mitochondrial and nuclear DNA sequencing because I just want to get an answer for them. Oftentimes by the time a patient has gotten to me, they've seen 4 other doctors and they just need an answer, and we'll just send everything.
So you said something off for mitochondrial DNA sequencing. Is it like a targeted, how do you make sure you get good enough depth of just the mitochondrial DNA?
Oh, you're going to get good enough depth because it's not done on the same platform. It's done on a different platform. So I don't know the specifics of it, but you actually have to order it separately through most of the companies because it's done with a different read depth. And like I said, you're going to detect variation down to 1 to 3%, which I actually wish we didn't get that kind of depth reports. Yeah, so this isn't like you're getting at, this isn't a yes-no like a Mendelian disorder. This is a yes, no, and at what percent. But you're actually bringing up a really, really good point, is this idea of heteroplasmy. What happens if you see a patient and they have a mitochondrial myopathy? Their muscles just start working, they're making a lot of lactate, but you sequence the mitochondrial DNA in blood and find nothing. What could be the situation here? What's possibly the problem?
The cells you're sampling don't have mitochondria.
Exactly. So there are some cases in which I'll actually biopsy, or my colleagues, I don't biopsy muscles, will biopsy a muscle to sequence that mitochondrial DNA to see if the variation is there. And we do find there's some variants that we know are more likely to appear only in muscles. So we do have to be very careful about the tissue that we're looking at. We also can't assume that the heteroplasmy that we're getting in blood is the same as what's in every organ system. So if you say get the 3243 classical mitochondrial DNA amino acid variation in blood at 30%, you can't tell the patient they're off the hook for the rest of their lives, even though 30% is low. You still have to say, actually, I don't know what's in your heart, I don't know what's in your brain, I think you're okay, but we're going to have to keep following you to keep you safe. And respiratory chain studies, I think historically these are really relevant when the way that we can diagnose mitochondrial disease is actually looking at the enzymatic function of the respiratory chain in a tissue. It's really non-specific. There's a lot of things that can affect it. It's really hard to prepare the tissue for it, but I still will sometimes use that to confirm findings. I think I'm using it less and less. There's been a number of studies to show that even if you send the same study to 2 very good labs, you could potentially get very different results. This is a diagnostic algorithm. I kind of just talked through it.
Did you say how what the incidence of mitochondrial disorders is?
It's about 1 in 4,000 people has a primary mitochondrial disorder.
Why is that surprising? Would you expect there to be more, considering how many mtDNA molecules, the likelihood of some?
That's interesting, Cody, because in my world, where most diseases I deal with are 1 in a million, 1 in 4,000 seems incredibly common. Yeah. But as I mentioned, the likelihood of finding a pathogenic variant in somebody, if you look, is 1 in 200. And the question is, if you look deep enough, are you going to find some disease expression there? Possibly.
So what's the kind of balance? Because you mentioned earlier, sometimes if you have a certain DNA mutation, depending on what it is, it might make the replication happen faster or whatever. So but what's the balance? So if a lot of folks have a pathogenic variant, what's keeping that pathogenic variant at bay, I guess?
I think randomness. I just think randomness, but there are some variants. I didn't want to get into the nitty gritty of this. If you really look close enough, at some point mutations, there is a replicative disadvantage.
Okay.
Really look close enough. And you can see
it's not as cut and dry as just saying
it's
they
not,
just have a
yeah, it's not, but it kind of helps us think about, in general, how we think about mitochondrial disorders. But I do think it's a numbers game. I think you have 99% of something and 1% of something, in all likelihood, it's going to stay that way.
Yeah.
So, for me, this is an important, I still talk about this every year. I've been giving this lecture for about 10 years now. I still think about this case because it's been really important for how much whole exome sequencing and now whole genome sequencing has kind of changed the game for how we think about diagnosing patients, but it's not the only thing. So I had 2 patients, they were siblings, who came to me probably about 10, 12 years ago with global developmental delay, muscle weakness. They had an unusual facial appearance. They had dysmorphic faces, which we don't typically see in mitochondrial disorders. Some slurred speech. The brother, who was a little older, had cerebral hemorrhage when he was born. He was getting a brain bleed, but he had a pretty traumatic birth, so it's hard to know what that meant for him, whether it related to the rest of the picture, and normal vision, heart, lung, and hearing. And when the brother was born, and he had that cerebral hemorrhage, they attributed everything to his brain bleed. Told the parents it's never going to happen again. And then they had the next child who had the same disease. So We wouldn't say that anymore, I think, with our sort of more knowledge of how to sequence and understand these things, but I guess 10 years ago our ability to detect this, but it's maybe a little bit different. I don't know, I'm being generous. I don't know. So the first thing we did in the siblings was SNP array, look for missing and extra pieces of chromosome, and it came back normal in both siblings. We sent biochemistry, so we sent amino acids. We found that there were markers for things that told us these patients had chronic lactic acidosis. They had chronic lactic acidosis, and broadly abnormal urine mitochondrial markers. So there's a sign of a lot of oxidative stress in their urine. We sequenced their mitochondrial DNA, and that was normal. So where do we go from here? Well, a couple of years after we started seeing them, whole exome sequencing became available. And GeneDx, I got to give them a lot of credit for this. They called out that each sibling had what looked like a pathogenic variant on a paternal copy of FARS2, which is a phenylalanine tRNA synthetase. Basically, it sticks phenylalanine onto the mitochondrial tRNA. Okay, so it's a nuclear mitochondrial encoded gene. They didn't identify a second variant, but they called this. And again, I got to give them credit because they didn't have to call this out. And so when we went and looked through the literature, there were only 3 cases of a severe infantile oxidative defect, and the patients all died shortly after birth. But we kind of got hooked on this, and so we started thinking about what could be going on in the other copy of FARS2 that we might be missing. And fortunately, since the SNP array was done here at Haskins, we called them and said, can you just take a look at the other copy of FARS2 and just, is there anything there? And there actually was an intragenic deletion on the other copy of FARS2 that was below their level of reporting. So they didn't call that because it was too small. It was 5 kb, and their level of recording was above 20 kb. And luckily, there was dense enough SNP coverage in that area that we actually were able to find that the maternal copy had an intragenic deletion. So they had FARS2 deficiency, and we reported them out. We actually wrote them up because they both survived. Unfortunately, the brother just passed at 25 or 26 a couple months ago, but the sister is still alive, and it was vastly different than what had been reported as an infantile death in these patients. So this really emphasizes that whole exome sequencing is great, but we still have other modalities. Genome sequencing wouldn't have missed this, of course, so that's why I think they probably have to start modifying this slide. So treatment, mitochondrial disorders. Right now, certainly for the mtDNA disorders, we cannot fix the mitochondrial DNA variation. CRISPR machinery doesn't get in there, so even though it's like a hot topic now, can't get in there. There's some people working on TALENT technology and zinc finger technology to get in there, and ASO technology to get in there and alter some of the variant expression. But right now, what we do is sort of, grossly speaking, try to keep the patients from having to make too much energy, right? So we just don't want to overtax their bodies. And what are things that tax the body? Fasting, right? If you fast, your body goes into catabolism, you start breaking stuff down. It's really stressful for your mitochondria. We don't want them fast. Fever, illness, anything that raises the basal energy expenditure of the body, we want to stop that. So sort of a principle of pediatric medicine is we let kids have fevers because fevers are fine and fevers help fight infection, but we do not let kids run fevers with mitochondrial disorder. We bring them into the hospital. We don't let them fast. We put them on IV fluids. And this is kind of a difficult thing with avoiding environmental extremes. You know, families call me and they're like, oh, my kid's not looking so good. We were in a hot tub. Yeah, they don't go in a hot tub. Or, you know, summertime when everybody goes to Disney, we get a lot of phone calls for kids not doing well. And they should go to Disney and love their lives, but it's hard on the body. There are things that we do to try to maintain basic energy supply. So, you know, supplying some cofactors for the respiratory chain theoretically sounds nice. In a petri dish, it does seem to help function in dysfunctional cells. I think the jury really is still out about whether or not this has a functional effect on patients, but this is all we got, so we give them antioxidants. We do recommend isometric exercise because this helps build up, you have type 1 and type 2 muscle. Type 1 is oxidative, type 2 is glycolytic. So we want to help them build up those muscle cells that rely on glycolysis. So isometric exercise, toning, things like that. And antioxidants when that respiratory chain, which basically functions as a circuit, isn't working well, you get oxidants escaping that causes lipid damage, lipid peroxidative damage, some membrane damage. So we do recommend antioxidants. It's sort of been a focus of a lot of therapeutic development. You know, I don't really know that we've seen positive outcomes for clinical trials, but 2 years ago, the first treatment for mitochondrial disease ever was approved. And this was for Friedreich's ataxia. Have any heard of this disorder? Yeah. So this is a repeat expansion disorder. So the Friedreich's ataxia people don't believe it's a mitochondrial disorder. The mitochondrial disorder people call it a primary mitochondrial disorder. They don't want to separate themselves from us. But they did speak at our last UMDF meeting, Mitochondrial Disease Foundation meeting. So I think they're on board. But essentially what it is when the frataxin gene has an abnormal nucleotide repeat, it impairs iron cluster biosynthesis. They get iron accumulation in the mitochondria, lots of multiple enzyme dysfunction, including in mitochondrial enzymes in the mitochondrial respiratory chain. So omoviloxolone was trialed in Friedreich's ataxia, which is thought to improve NRF2 signaling and decrease NRF2 mediated and improve NRF2 mediated antioxidant response to the stress from the iron sulfur clusters. Okay, so what they're basically trying to do is activate NRF2 to decrease oxidative stress caused by these iron sulfur issues. And I don't know the best way to phrase this. I can't remember, Julie and I used to talk about clinical trials and how to design them. I don't think we do that anymore in this class. But what's really separates out the basic science from how you get it approved is you can have a mechanism and it can look great in the dish and you can have all the markers you need, but you actually need to show that there's an effect in a person, and that effect doesn't have to have anything to do with the mechanism. Does that make sense? Like, you can say why you think is happening and what you think is happening, but you actually have to show that the person is physically getting better regardless of what the mechanism is. Does that help? It's tricky. It's just a tricky concept. So what they actually did was looked at, they put patients on omoviloxolone for a period of 48 months and compared how they did in terms of their Friedreich ataxia symptoms compared to natural history. That's how they got this drug approved. It wasn't based on mechanism, it was based on this outcome. And so this was approved just a couple of years ago, pretty recently. And has, I think, been a real boon to morale for the fact that, you know, in rare diseases and mitochondrial diseases, we can get things approved. So this was developed at CHOP, now widely available. Okay, I think I got through it in 65 minutes. Okay, what questions do you have?
Do you know if mitochondrial transplant is becoming something that would be used in people? I know it's very flashy. Right?
So there's two clinical, periclinical applications right now. So the first one is, and I hate the term 3-parent embryo. This is probably how you've heard of it all. Is this idea that for mothers who are carriers of mitochondrial DNA variation who want to have biological offspring but don't want to pass along the mitochondrial DNA variant, you can actually replace the mitochondrial DNA, or the mitochondria in the egg, and so you can transfer just the nucleus from biological mom, the mitochondria from somewhere else, do IVF to marry that with the sperm, and then have an offspring that way. That is not something that's legal in the United States currently, but the UK has mechanisms for this, so sometimes families will go there for that. There is a company right now, I can't believe their name is slipping my mind, that is doing mitochondrial transfer in Pearson syndrome and other mitochondrial deletion disorders where they take, it's essentially an in vitro, it's not even really bone marrow, so they take out hematopoietic stem cells from the child. They take mitochondria from the mother, because these deletions, again, are not inherited. So, right, so the mom has the same mitochondria but without the deletion. Does this make sense? Should I draw it? Y'all got it? Okay. And they basically put it in a dish together, and the child's hematopoietic stem cells take up that mitochondria, because we do know that there is mitochondrial transport from media and things like that. They put those stem cells back in the child like a bone marrow transplant, and they do clinically do better. So currently, those are the two real clinical applications that are going on. Now, there are people working, like Mandeep Singh in ophthalmology is working on retinal mitochondrial DNA cancer. He's got these really cool images. If you ever have the opportunity to hear him speak, it's really neat. Like the retinal cells will send out tubes to other retinal cells and transfer their mitochondria between each other. And he's trying to take advantage of that as a therapeutic for retinal disease, but I don't know that that's clinically, like, that's out there in the market yet.
So what is it about, like, what special thing happens to mitochondria in the mother that makes them compatible with the egg? So when you do a 3-parent, why can't you just take mitochondria from the father and put it into the mother's?
Well, where would you take it from? Right? You want to make sure that you're keeping cell type to cell type, I guess.
So I guess what's unique about, like, ovum mitochondria versus...
Well,
like
the answer to that.
Oh, okay.
Yeah, I don't know the answer to that. All it is... There's got to be something special. There's some reason that that's why they're doing it.
Sure.
Yeah.
For patients that have mitochondrial disease, even if they're doing like okay, like not necessarily Q6, will they still have like a high lactate?
It's hit or miss, which is, from a clinical standpoint, which is why when patients come in and they're in clinic and they're well, why I always measure lactate at every outpatient visit, because I kind of want to know where they're at when they're doing well, because it really bounces around unpredictably. But they can, when they come in and they're sick, those levels can double, triple, quadruple. But sometimes the other thing I'll do is have a patient where I'm not sure what's going on, measure a level and have them walk around a lot, get some things active, and then measure again. But plasma lactate variation is super tricky because it does go up and down, even in patients with severe disease. If you do a spinal tap and look at CSF lactate, that's actually much more measured, much more the steady state. So I will always believe in elevated CSF lactate. That's always real. Nothing else. Well, you all know how to find me. I think I provide a copy of this, right? Do I provide a copy? Yeah, so I'll send out a PDF of this. Any questions, thoughts, concerns, just come find me.
All right, thank you.
I'm going to get applause after I teach.
The latest and final schedule for the rest of the class is posted on campus. We are still expecting Eric Green to give the last lecture in the class. As far as I know, he's still coming. And the last class will be the last discussion session, which will cover mitochondria, epigenetics, and induced pluripotency.
Yeah, well, I was just gonna say we could just do the when I get back in the room, I'll pull up campus.
So, for example, we talk about like exposure change. There's one that were 47 subunits. 40 of them are actually in the nucleus. So that makes our primary mitochondrial disease, which is a defect directly of the gene encoding one of the subunits of the change. Does that make sense? Or if you think about, mitochondrial fusion and fusion, that helps the mitochondria. Yeah, and those are included in the nucleus, so it still affects the way the mitochondrial primary
It's like Vasilides, like her holding, so you just go by that, so.
Yes.